Okay, cell nucleus. Most everybody has, and when I say most everybody, most cells have a nucleus. Most cells just have one nucleus. The two exceptions would be muscle cells, skeletal muscle cells, I guess I should say. I'll put SK for skeletal. Skeletal muscles are usually multinucleated. They can have more than one. And red blood cells, mature red blood cells, have no nucleus. So obviously those are the exceptions to the rule, but everybody else has at least one. When we look at the cell nucleus, there she blows. It's just a highlighted, blowed up version of the cell nucleus. All of that is. It is a double layered membrane, just like the cell membrane is. We have tiny little holes within the nuclear envelope, nuclear plasma membrane. Let's see here. We have a nuclear pore. Nuclear pores are there to allow the messenger RNA and the small ribosomal subunits from leaving the nucleus. Ribosome subunits are made by the nucleolus found within the nucleus as well. So all of those things have to leave the nucleus. They leave through those tiny little pores. And again, there's that nuclear pore right there. Okay. There's the nucleolus. Makes ribosomal subunits. Okay. And then there's the uncondensed genetic material. All going on right there. Nucleolus disappears, nuclear membrane will fragment and disappear during the whole mitosis part. As the cell starts to divide, that chromatin will start to condense down into chromatids, TIDS. But yeah, those are all the parts and the pieces for the nucleus. It has all the little machinery inside of it to direct the functionality of the cell, i.e. the genetic material. That's the chromatin, okay? Excuse me. Excuse me, guys. We have two parts when it comes to DNA and genes and understanding how we get from DNA to a protein. We have transcription and translation. Where do these occur? First one occurs within the nucleus. Second one occurs in the cytoplasm. Okay. For transcription, that's where we just make a copy of the code. Okay. We copy the DNA and package it up as a messenger RNA molecule. It leaves through that little tiny little nuclear pore and goes out into the cytoplasm. Once we're out there, that messenger RNA will dock with ribosomal subunits. and we will read it. When you read something, you translate it. And that's what translation is. Okay. Ribosomes plus messenger RNA yields the translation process. This is sometimes referred to as the central dogma of biology. So if you've had 101, that's probably what it got described to you as. Okay. It's a major concept of biology in the grand scheme of things. Okay. This transcription translation process, how we get from DNA to an actual physical protein. Okay. When you make the protein, it's referred to as being expressed because now we have a manifestation of something. It's expressed. So that's where that whole gene expression or central dogma of biology kind of occurs. Function. Making the ribosomal subunits is the function of the nucleolus. Really not very spectacular. I'm gonna do this. Don't worry about the genome and the proteome. The genome is the totality of all of your genes in your DNA. I'm not going to get into it, so I'm going to cross it out. So that means don't worry about it. All of the proteins that the DNA codes for, that's referred to as the proteome. There are subsections of research that look into all of the genes and all of the sequences to see what proteins those genes code for. There is a growing body of science that's looking into the actual physical proteins that are made, how those can go back and affect the genome and affect their ability to make more of those proteins. I don't really want to get into it any more than that. I'm not going to ask anything on the exam. The authors go into it a little bit. If you're curious, there's a ton of research out there on proteomes or whatnot, and how nutrition can affect the proteins that are made and what. There's some interesting stuff out there, but these two slides, guys, don't worry about it. Okay, don't worry about it. I'm going to highlight a few things here. When DNA is loose, it appears as just a bunch of clouds, right? It's not condensed down. It's not thick. We see that under a light microscope as just kind of this cloud of material within the nucleus. When DNA starts to undergo the process of condensing down, we need to get this stuff to be wound up. Okay, this genetic material, these long strings of genetic material needs to be wound up. To wind those up, we wind them up around some small little protein bodies that are called histones. And here's what they look like. Okay. And here, all of these histones, they just give material for the DNA to be wrapped and condensed down. Okay. When chromatin starts to condense down during mitosis, let's say, all of those histones start to get wound up with genetic material, chromatin. This condenses down into those X's that you see on the slides. When you can see the actual chromosome, they are made now of chromatids. This is condensed genetic material. Okay. Wish I could spell today. Okay, so that's the condensed genetic material. Chromatin, chromatids. This stuff is all a review, and the most common things screwed up is this thing right here when it comes to DNA. Okay. Chromatids are the condensed version of this genetic material. The uncondensed version is referred to as chromatin. A chromosome is comprised of 2 chromatids, and they're referred to as sister chromatids. That spot right there in the dead center that is the centromere, and that's what holds the 2 sister chromatids together. Where the spindle fibers come down and attach to and move this little chromosome around during the steps of mitosis, which we'll talk about briefly. Okay. When we look at the condensed genetic material, there is a ton of information there. And like the vast majority of it actually codes for nothing. It may have coded for something, but it doesn't code for really anything. It's inactive, okay? It can't be read. There's so much stuff in that region that it's not readable, okay? The stuff that does get read, the stuff that does get interpreted is the U chromosome, or the U chromatin, sorry. Okay. This is the stuff that gets read. This is the stuff that was ultimately going to be expressed. On the exam or on the quiz, either one, I am not going to ask you to write out the steps of transcription or translation. I am not going to do that as a short answer question. I got other questions to ask for that. But could I ask for the basic steps like I have listed down here in terms of a multiple choice type question? Sure. And that's what I generally do. Okay. That's what I generally do. Okay. When we look at how we transcribe and translate, they are both basically the same. Okay. They'll have a number of starts and stop regions. There'll be regions that are going to promote the reading of the DNA material. There's going to be regions involved with stopping the reading of the genetic material. So there is a lot of commonality between the two. And I've tried to keep it super duper basic and simple here. Okay. When it comes to transcribing the genetic material, we have what's called DNA polymerase that will unzip the DNA, open it up, and allow for a transcription bubble to occur. Okay? We always start reading the DNA at one location. Okay? There is only one start spot. Okay? And as long as I have those 3 sequences of letters, that's where I start the read. When it comes to stopping the genetic reading, there's usually 3 different codons, 3 different amino acid sequences that we're looking for to stop. So there's always one start, but there's multiple periods within the sentence. Okay. We have that DNA polymerase. That's where we unzip this sucker, open it up, and start copying the material down. Okay. And there are a number of other enzymatic functions that are going to be involved with this thing as well. I'm highlighting important things that I would generally ask on an exam, things that you would see as a distractor, as a choice in a multiple choice type format. Okay. So I have DNA polymerase that opens up. I have various different transcription factors that come on in and allow this reading of the DNA molecule to occur, stringing together my individual amino acids, complementary base pairing, A with T, G with C, or A with uracil in the RNA case here. Okay. And we copy one strand of that genetic material, that one DNA side, and we send that out into the cytoplasm as messenger RNA. Okay. If you read the book, it goes into the pre-mRNA. What is pre-mRNA? Before it leaves the nucleus, guys, I have to manipulate the thing a little bit. I have to put a little cap on, I have to put a little tail on it for it to be removed properly from the nucleus, for it to be delivered to a ribosomal subunit properly. Okay. And that's all that pre-mRNA is. Okay. So as soon as we read this thing, I have to put a little poly A tail on it, a little cap, and now this messenger RNA molecule leaves. I have transfer RNA. Transfer RNA is found only in the cytoplasm. Transfer RNA brings specific amino acids to the ribosome. Which tRNA molecules come to the ribosome is determined by what we read on that mRNA strand. The other type of RNA that we have out in the cytoplasm doing stuff is ribosomal RNA. Ribosomal RNA only does one thing, like I mentioned earlier, I believe, when I talked about ribosomes. They form the ribosome. They form the small and the large subunit, okay? And at that ribosome, that's where we translate what we have written down. Where we make the donuts. Okay. So here's a fabulous picture here of looking at how I've unzipped DNA, opened it up, and allowed one strand of this thing to be copied. As this messenger RNA that I've formed here leaves, it needs to be modified. There's my pre-mRNA. Okay. I put a little poly A tail on it, put a little cap here on the end of it here. And I've also removed all of the regions of the genetic material, those heterochromatin regions that have way too much information on it, and I remove them. Okay. When they're in the pre-mRNA molecule, we refer to these regions that code for nothing as an intron. There's actually some current research as to looking at what an intron actually, why is it there? The vast majority of the pre-mRNA molecule that is manufactured actually doesn't code for a damn thing. So we have a ton of potential genetic material that actually doesn't code for anything currently in a human. So these introns have to be removed for the translating of the protein to be done properly within the cytoplasm. What they're finding with these introns is kind of interesting, is that if for some reason the cell reads the DNA wrong, or reads this pre-mRNA molecule wrong, then all of a sudden that intron becomes active. And it's like a switch. And it codes other proteins within the little exon segments to turn on or off. And they can actually, they know a few of these introns and they know a few of these switches, is what they call them. And these switches can, in fish, there is a couple different fish species within the same family where the intron has been expressed and it produces these tiny little spindle-like fins on the bottom side of their belly. And there is a very similar fish in a different location, isolation, separated from it, where that intron is not switched on. And this fish does not have that same spindle-like fin on the bottom side of their surface. So they're actually kind of looking at these introns in terms of these genetic potential markers, if you will, for how evolution has kind of happened over the millennia. And it's pretty interesting. They've discovered these switches for how arm buds form in certain animals. It's pretty interesting. And this is only within maybe the last 20 years or so that they've been able to kind of see these. So these introns, even though technically do not code for anything specific, they may end up being these switches that can turn on and off during misreads of DNA, and something that's on can all of a sudden be turned off, and something that's off can all of a sudden be turned on. And that gets a whole different read of the genetic material, and that gets a whole different outcome to where you can have a new species emerge if enough isolation occurs with that animal group. So they're pretty interesting, but introns themselves, they don't code for anything. They're actually junk DNA. But when in terms of from an evolutionary standpoint, they've done a lot of stuff with this. Again, supercomputers, knowing the genome of various different animals, that has obviously kind of helped bolster our understanding of some of this stuff. But it's pretty interesting. Am I going to ask you anything about that? Probably not. I just find it very fascinating and interesting that these are swaths of DNA that code for jack nothing. They code for nothing. But all I got to do is misread this sucker right here, and all of a sudden, something in this region becomes active, and you get an entire different read on the genome. And I don't know, to me, that's pretty fascinating, aside from them not doing a damn thing.